Okay. Good morning again, everyone. I am Ajay Pillai. I am a program director at NHGRI and part of the team that manages and oversees the Illuminating the Druggable Genome program. And today I'm here to talk about the new CEIT RFA and then associated NOSI that was published recently. Some general announcements. So this presentation is being recorded so that we can make the video available for people who couldn't join or for all of you who wanted to listen to a bit again or whatever be the reason. So that's announcement number one, so that this video is going to be made available at the Common Fund IDG website. The second announcement is that you can use the chat function and you can select any of the host or co-hosts that you see listed and text them any questions that you have. You're of course free to ask the question using the microphone as regular in these Zoom meetings. And those are the two general announcements. And I'm going to start screen sharing and start my presentation. So I assume that you can see my screen. So as I mentioned earlier, this is a pre-application webinar for RFA-RM-21-020 titled Cutting Edge Informatics Tools for Illuminating the Druggable Genome. It's a UO1 and there are no clinical trials allowed. Towards the end, I will also talk about an associated administrative supplement, NOSI.
NOSI is a Notice of Special Interest.
That is about getting single cell data into the IDG knowledge base. So I mentioned that we can take questions and address them, and that there'll be a transcript made available. So let me start off by telling people what Common Fund is. Common Fund is a particular, well, it's not an institute, but it's part of the Office of the Director, and they do a number of projects, and they are unique in many ways within the NIH. So generally speaking, Common Fund programs are expected to be highly transformative. They are catalytic for getting certain new technologies and new approaches up to a certain level, and then they're expected to be just picked up by individual ICs or through other NIH mechanisms. So they are short-term and they're goal-driven and they last for 10 years. They are also synergistic and enabling so that they advance the missions of multiple NIH institutes. So for example, they're not just simply cancer-focused. And they are cross-cutting. They address complex issues that require trans-NIH teams. And they are novel. They provide new solutions to very specific challenges. So those are the general features of all Common Fund programs, and the Druggable Genome program fits within that framework. So this is just a bit of history to take you through why this program was set up as it was. So these are graphs that talk about the proteins within the GPCR ion channel and kinase families. where a very large number of them are understudied. So there aren't many publications about it. There aren't many reagents and resources available for them to investigate what they do within biological systems. And the IDG program's basic goal is to ameliorate the situation so that a lot of the understudied GPCRs, ion channels, and kinases can be studied more. So that's the overall goal for the IDG program. This is a similar thing as in the previous screen, again, talking about GPCRs, but the story is true for all 3 protein classes and likely other protein classes as well. But the other protein classes are not the primary focus of the IDG program. So as mentioned, this program was started in 2014. There was a pilot phase for the program, which lasted about 4 years, from 2014 to 2017. And we are currently in the implementation phase of the IDG program. And the general aim, so as background, these slides have a lot of material. I am not necessarily going to talk about everything on the slides. A lot of it is for people to poke around later. So as I mentioned, in the goals of the IDG program are to identify biochemical, cellular, animal model phenotypes for understudied proteins from the 3 protein families, so that the community can investigate these proteins by providing reagents and tools, and also to generate and maintain a mineable knowledge base for these understudied targets. I wanted to give, so if you recall, this is a UO1, which means it's a cooperative agreement. And so you are joining sort of a consortia. The CEIT awardees will be joining a consortia. And it's useful to know what the structure of the program is and how some of these things are maintained and done. So the general structure of the IDG program is shown on the left. There are the experimental groups that are the data and resource generation centers. So these are the groups that generate the tools and identify various biological role for the understudied targets. We also have a resource dissemination and outreach center. So as you recall me saying before, we want a lot of the understudied targets to be studied further in the community, and we have an outreach and disseminations center that is focused on doing that. There is a knowledge management center, and there's a specific web browsable tool called Pharos, and these resources give you access to a large amount of data in a usable manner focused on studying any of the proteins that the IDG program is interested in. As I mentioned, the cutting edge informatics tool, we are here to talk about this RFA. This is essentially a reissue of this RFA that was done in 2018.
The RFA is for the Cutting Edge Informatics Tools, or CEIT awards.
We have a large number of RO3s that have been issued every year, and there is one that is currently out on the guide, and that talks about generating preliminary data and experiments to create new assays and to study the understudied targets within the IDG space. We also have SBIR and STTR active PARs and announcements, funding opportunity announcements. In general, you can go to the Common Fund website for IDG and look at the grants.
The website is commonfund.nih.gov/IDG/grants #current.
Just a bit more about the data generation centers. As I mentioned, there are 3 target families, and there are 3 groups that are focused on the target families, GPCRs. The experiments groups are Brian Roth and Brian Schreikert. Protein kinases is under Gary Johnson. And for ion channels, it's a collaboration between Lily John and Michael McManus. As I also mentioned, one of the things that we really want to be able to do is to generate data in these experimental centers, experimental labs and groups, and to create tools. So here's an example of one of the tools that has been published. I'm not going to describe a lot of what you're seeing, but you can check out the publication.
The publication title is Allosteric Ligands for the Pharmacologically Dark Receptors GPR68 and GPR65.
The Resource Dissemination Center is a collaboration between University of Miami and University of New Mexico, and they manage the IDG website.
The website is druggablegenome.net. The listserv email address is idg.rdoc@gmail.com. The Twitter accounts are @druggablegenome and @IDG_Pharos.
And there you can find the status of different proteins and gene families within the IDG spectrum, and how much illumination, how much work has actually happened within the IDG consortium. So you can see, for example, that we work all the way from the cellular and model organism levels. As I mentioned also, there's a knowledge management center that maintains a very large database. This is a database that is extracted from many other resources on the web. That's publicly available, is organized in a manner that is friendly to biologists. It is under constant evaluation and improvement. And we have a web-based portal to access all of the data. There's a very large amount of data. You can access the KMC products through Pharos. There's a URL up here.
The URL is pharos.nih.gov.
There are many ways you can do it through browsing and filtering on the website, and you get very specific and detailed views of the protein that you're interested in. And you can also access parts of the underlying database through APIs, or you can download the entire database if you want to do large-scale processing of the data sets. Okay, so generally speaking, so this is the RFA.
The RFA is RFA-RM-21-020.
I'm going to switch now to talk about the RFA of interest right now. If you have any questions, please email us at this email address, thedruggablegenome@mail.nih.gov.
The program contact is Jerry Lee. The grants management contact is Krista Wolfrey. The email address for Krista Wolfrey is wolfreyc@mail.nih.gov.
Some key dates about the FOA. So the FOA is open for submission on June 15th. It's generally useful if you let us know that you are going to be applying through using the letter of intent. It is not necessary to do this, but it helps us organize review better and gives us more time to plan out various things. The last date for the applications due is July 15th. The scientific review will happen through a special emphasis panel that's organized by the Center for Scientific Review, and it'll happen sometime in November. These awards are, these applications are going to go for advisory review in January, and the earliest start date is going to be April of 2022. So general administrative details, and again, these are not your standard UO1 applications. I want to repeat that. I mean, sorry, standard R01 applications. So it's important for you to read the FOA carefully. So for example, there's a resource sharing plan. You should go through that. There are the usual authentication plan that NIH now requires in almost all applications.
The website is grants.nih.gov/reproducibility/faqs.htm.
There is also the consideration of sex as a biological variable, which is again a standard now requirement for NIH funding.
The website is grants.nih.gov/grants/notice-FOD-NOT-15-102.html.
Your research plan, there's a 12-page limit. There is also a data sharing plan. And again, you should read the data sharing plan carefully, that you understand what is expected of you. And of course, the usual caveat in NIH applications is that please don't use the appendices to circumvent page limits. Coming to the scientific review, these are, again, UO1 grants, and they do not have standard R01 criteria. So it's important to read the review section of the RFA, because there are a number of questions that are in the review section that pertain specifically to this FOA. So, like I mentioned, the SRO is going to be Dr. Vinod Charles from the Center for Scientific Review.
The email address is charlesvi@mail.nih.gov.
Generally speaking, again, and this is important, we are likely going to apply a number of criteria to make the awards, and it is not just going to be necessarily done in, for example, score order. So some of the criteria, I mean, these criteria are listed in the FOA. I wanted to highlight some things out of this longish list. One is that as a program, we are interested in expanding out to the community and getting new investigators involved. This is generally speaking a very important criteria for most of these sorts of programs that NIH manages. And the other main thing that I wanted to add is that diversity of the technical and intellectual approaches that people bring to the IDG consortium is also going to be an important element in making funding decisions. I mentioned earlier that the resource sharing plan, we should pay attention to it. The primary goal of the IDG program is to make data and resources readily available to the broad scientific community. That means if you actually have, for example, you are required to share data, resources, protocols, tools, software in a way that will achieve these goals, right? So all applications must include a resource sharing plan, and the applicants must indicate their willingness to abide by all of the data deposition, quality control, standardization, metadata requirements, data and software releases, and public copyright license policies that are generated by the consortium. A lot of information about about Pharos is available on the Pharos website.
The website is pharos.nih.gov/IDG/FAQ.
If you have any specific questions that you want clarity on either the Pharos or IDG, you can always send an email to us at the Druggable Genome thing, and we'll be happy to answer your questions. And again, as I mentioned before, please, you will all be generating software, and it's important for you to read the software sharing plan in the FOA.
The website for the sharing plan is grants/rfa-files/rfa-files.rm-files2one-020.html/section_application_1.
This in the next slide is essentially going to be a bunch of URLs.
The URLs are commonfund.nih.gov/IDG/webinars for the webinar URL. Druggable Genome at mail.nih.gov for the help email. druggablegenome.net for the consortium homepage. And druggablegenome.net/IDG resources table for the IDG experimental resources website.
Again, like I said, this presentation is going to go up on our website and will be usable in general. I'm not going to talk about any of these in specific detail.
Websites listed include pharos.nih.gov for Pharos, pharos.nih.gov/API for Pharos REST API, pharos.nih.gov/FAQ for the general help page, pharos.nih.gov/about for data sources, juniper.health.unm.edu/tcrd/download to download Pharos data, bit.ly/IDG YouTube for IDG YouTube channel, and druggablegenome.net for the IDG website.
So out of this RFA, we generally expect to make about 3 awards. There's a total of $1.4 million available for this FOA that has been set aside. However, an individual application there's a maximum of $300,000 in direct costs, and the awards will be made for a period of 2 years. So generally speaking, what are the goals and approaches and strategy for the CEIT applications? So there are 2 sort of ways in which the FOA conceptualizes the applications. So you can either get into the space of providing predictive models. So within biological context, one of the things that the IDG does is try to provide the community which are the targets that are more likely than not be involved in a specific biological process or in a specific disease or in a specific cellular validation, so that you can set up experimental validation methods appropriately, and you can study and choose which of the understudied proteins you want to investigate. So in general, one of the goals of the KMC is to enable such prioritization of proteins within specific biological contexts for investigation. Like I said, it could be a biological process, a disease, or a pharmaceutical relevance. So that's the overall goal of the Knowledge Management Center and Pharos, and the CEIT FOA is essentially there to help build such predictive models. You can think of predictive models as ways by which you do molecular machine learning or AI or any of the other ways in which you can build predictive models.
The website for making models available is pharos.nih.gov/FAQ.
You can also build predictive models by allowing people by constructing various visualizations that reflect prior knowledge and be able to enable the user to think up new hypotheses. So these are the two ways in which the FOA conceives of what sort of approaches an applicant can take. In general, one of our experiences has been that ion channels have been particularly hard to get innovative and useful predictive models and other types of visualization and user interface design questions to prioritize. And we are looking forward to getting innovative and interesting ion channel focused experiments. But in general, your application can target any of the three classes of proteins of relevance to the IDG. Again, like I said, there are specific review criteria questions. Oh, I forgot to mention that there is a requirement to do some amount of experimental validation, especially if you build a model. And these sorts of validations are just preliminary, just to give you an indication that your model is not completely way off. And, we are not specifying, the FOA does not specify a number for what percentage of budget can be used for doing experiments, but we do not intend this FOA to be focused on primarily experimental approaches. So use your judgment in how you would undertake experimental validation. Coming back to the review criteria, so significance questions address things like, are you likely to make accurate predictions about the biological role? Is the approach likely to make? And is the proposed experimental validation suitable for generating preliminary but not deep experimental validation? Are the data sets that you choose to build your models off of appropriate and justified? And if you are proposing to use data sets that are not currently available within the IDG resources, are you proposing to make them available to the community? Then there are issues about things like containerization, provenance issues, and fairness issues about your approach. And finally, your tools are supposed to be deployed essentially in Pharos. And the Pharos team usually works with all of the members of the IDG consortium to enable these sorts of deployments. And you will see some of these tools deployed on Pharos currently. Okay, that brings me to an end about the CEIT FOA. And as I mentioned, I'm going to say a few things about the administrative supplement opportunity that exists. And this is specifically focused on getting single cell data into the IDG knowledge base.
The NOC is for NOT-RM-21-019. The contact email address is druggablegenome@mail.nih.gov.
So currently, there is a very large number of data types and data sets that are available within, that are accessible within Pharos. And we have visualizations and the ability to interact with each of the types of data. However, we are lacking in any single cell data within IDG. And single cell data is quite new, and it requires very specialized methods to analyze. And what we are looking for here is through an administrative supplement. And this application is due June 15th. Here's the notice. There's an important element that you need to mention NOT-RM-21-019 within your application box 4B in SF 424 form. So please don't forget to do this. The budget is $175K direct cost per year and this is for 2 years. This will go through an administrative review.
The slide states, we strongly suggest applications are submitted a week in advance.
Okay, the key goals, as I was mentioning about the NOSI, is that, for the application for supplements to active grants, so help process publicly available single cell data for query and presentation for use by biologists on Pharos. So the general idea is that you will establish a pipeline for processing, and you will think about what are the best ways to query single cell data within the context of enabling the prioritization of which targets to study under which biological situation or constraint, and also think about how best to present these single cell data to the community. So, I mean, the processing would essentially end up creating, something like a gene by cell matrix, and this requires specific expertise in being able to, having dealt with single cell data. And we do not specify, the vast majority of single cell data is essentially RNA-seq based. There are some single cell data that goes beyond and looks at proteins and metabolites as well. So, and essentially the innovation parts of your application is essentially going to be looking at how you query and present the data for analysis. And one of the other requirements for any processing of single cell data is the ease of maintenance for the resulting software. So with this, I come to an end for the presentation and open up for addressing any questions. So I think one of my colleagues, if you can ask questions, or if you prefer to type it into the system, one of my colleagues is going to ask it for you. And
so far we don't have any questions in the chat box. I don't know if they can ask questions in via the microphone. I think that if they're not panelists, they're muted. So they may have to type in the question in the chat. Okay, we do have one question. Are the slides going to be—
Carly, I can see the questions. The question
okay.
is, are these slides going to be posted? Yes, they will be posted this week, I hope, or if not, early next week.
Okay, I'm not seeing any other questions at this time.
So the next question is, preliminary data need to be generated during the course of the project? Does it have to be already completed? Well, so if the question means preliminary data in terms of whether your computational method is likely to work, I think in general you should consider this as, a standard way in which there is some amount of preliminary data that your approach is likely to work in some context. Without that, it's going to be hard for the reviewers to judge. So, but it does not need to be demonstrated that it is useful within the context of prioritizing understudied targets, if that makes sense. So the next question is, could you elaborate on what kind of computational tools you're looking at to bring out the biological relevance? And we are agnostic. Be creative. So usually, right? I mean, if you, what you should, one way to think about it is the overall goal of the IDG program is to get some of these understudied targets worked on more in the community, right? So to get biologists to start doing experiments in that, right? So in some ways, you should think about, are your methods likely to be evidence that somebody can put into their preliminary application that says, hey, here are some computational approaches that predict that this particular protein has a particular function in a particular cellular context, and they can use the models that you are building to put in new applications or start a new hypothesis-driven experiment. I invite my colleagues to, my colleagues at NIH, to elaborate on these answers if they think that something that I'm saying can be said better.
Hey, this is Carly Sharma. I'm the IDG coordinator. I think you're doing a great job, Ajay. I'll just mention kind of a little bit more on what you just spoke to. That the ultimate users for Ferris are going to be biologists, researchers, that type of thing. So whatever you're developing or proposing for this funding opportunity would need to keep that in mind as you put it together. What is going to be useful to that particular community? So that's all I'll say about that. But I just want to make sure that everyone's aware that, the ultimate user is typically going to be biological researchers. So just keep that in mind as you're developing your projects.
So I'll say that in a slightly different way. I think the FOA describes the goal for both the IDG program and the CEIT efforts. I think it's useful to think about whether the IDG goals are going to be met. It's actually a specific question in the review too. So you should address that, how your proposed approach is going to meet the overall goals. So the next question is, I am new to IDG. Do we need to show any evidence of collaborations with existing IDG awardees or with Ferris? No, you don't. I mean, if I said that, I think it creates one question like I said, there is a lot of information about, the software technology stacks that are used in Pharos. If you are thinking of deploying, some complicated visualization, it becomes important for you to know that, you're working within certain frameworks of software engineering and what is possible and may not be possible. I think we have, so you should feel free to ask specific questions about Pharos, either now or later. If you ask me now, I would likely have to get back to you later, depending on what level of detail it is. Next question is, what level of so it's the same question. What level of familiarity with Pharos architecture is expected? So again, let me repeat, I think you should go and read the FAQ and Pharos, right? It tells you general things about the software stack that is used, how it is approached. There is going to be some amount of flexibility. I mean, there are many ways you can skin the software cat, and talk to us in more specific details if you have any specific questions. But yes, I mean, you should show some amount of familiarity in your application with what's in Pharos. And I'll add that, I mean, a lot of what's in Pharos is very modern in terms of the approaches that are being used. These are not 1990s technologies, just.
We haven't received any questions for druggable genome inbox either, just let you know. That may be it.
Okay. I forget what our policy with early ending is. Do we just stick around for the required one hour, or?
Well, a question did just come in. We can answer that, and then yes, I think maybe we will stick around a little bit. Might be good in case others have questions.
So the next question is, are there any specific disease areas of interest to IDG? And the answer is no. The only, so let me say this, right? I mean, a lot of computational methods that are going to be fairly general, right? And a fair number of them can actually be used immaterial of diseases and immaterial of even target classes, right? You can build models that would just be able to predict on a very large range of protein families. We recognize that. I think I would recommend that the experimental validation that you should perform should focus on the 3 target families that are of relevance to IDG, and not other target families. So that's the only constraint there is, but there are no disease area constraints. Any more questions? Anybody being shy? If you are shy, just send us an email on druggable genome. or you can send us a question even if you can't think of one now, and you can think of one later. And I mean, in general, either Jerry Lee or I, and we can set up appointments to talk to you one-on-one as well. I think we've done that once or twice already. So if that would be helpful, please reach out to us. Okay, so I think I'm going to bring this to an end now. Thank you all for coming, and look forward to some interesting applications. Okay.
Thank you,
Bye-bye.
Ajay.